Hello everyone. In this session we will talk about DNA replication. So again, this is the reference book that I've used. You can get this book from the OpenStax. It's a free version, so you are welcome to go back and review this book. So let's talk about DNA replication. Okay, so what is the DNA replication? DNA replication is a process by which the replicative enzyme use the template strand and they use the template strand to make the new identical daughter strand. Okay, so this is process of just making a new DNA strands from the existing strand. Okay, and as you already know, and we will talk about later about the cell cycle. And in order for a new cell to make, in order for a body to make the new cell, it has to replicate the full genome. Okay, without replicating the genome it cannot make any new cell. Okay, a genome must have to be replicated. Okay, and there were different types of model that were proposed back then, and there were like three models that was suggesting about how the DNA replication occurs. Okay, and one model was a conservative model, and that model suggests that the new strand that is made, it does not have any parental strand. Okay, so for instance, on the right side of the image, as you can see here, this is the conservative model. Okay, and as you can see here, the new strand that is being synthesized, it does not have any parental strand. Look at this strand here. So this is totally new strand. This is a totally new strand, and it does not have any parental strand. The second on the middle image, this model suggests the semi conservative this model is the semi conservative model. And this model suggested that the new strand that is made, look at this strand here. So the new strand that is made, it has a parental strand. The the gray strand that you can see, this is the parental strand. Okay, so one strand is a parental, and the newly synthesized strand is a daughter strand, which is a complementary to the parental strand. This is the semi-conservative model. And there was another model, which was called a dispersive model, and this model just suggests that the newly made strand, it has the evidence of both parental and the daughter strand, but they are occur in the pieces, as you can see here, this piece here. Okay, as you can see, the one piece is a newly made strand, whereas the other one is parental strand. The semi-conservative process, it suggests that the newly synthesized strand, it is complementary to the parental strand. Okay, and and this is something that I have here already mentioned. The parental strand serves as a template strand to make the new complementary daughter strand. And again, the newly double strand contains a one parental strand and a one daughter strand. As I already mentioned here, the gray one is a parental strand, and the blue one is a daughter strand, which is complementary to the parental strand. Okay, okay, so DNA replication is a very complex process. Okay, in eukaryotes, we will talk about the difference. Like the process is relatively same in both eukaryotes and prokaryotes. Just the naming of the enzymes are different. So I will just I would just use the prokaryote as an example here, and you can obviously and I also put the naming of the enzyme for eukaryotic at the end of the slide, so you can go back and look at that slide. Okay, so in eukaryotes, there is a the replication process begins with the multiple origin of replication. So recall that the eukaryotic DNA is a linear and they are long. Okay, so it makes sense of having a multiple origin of replication. So the eukaryotes has multiple origin of replication, which allows them to replicate faster, because having a multiple origin of replication allow the DNA to synthesize simultaneously. Okay, whereas in prokaryotes there is a only single origin of replication, because prokaryotic DNA is circular, so, and it is small. So having a multiple site does not make sense because the genome is very small. Okay, and the replication process begins with the initiator protein. So these initiator proteins, they actually exposes the DNA to the replicative enzymes. Okay, so here on the right side of the image, as you can see, these all replicate or initiator proteins, uh, like single stranded protein, helicase, this is all enzymes. So they actually expose the DNA to these replicative enzymes. There are other initiator proteins as well, but it's not relevant to know. But these proteins are actually attract the DNA. DNA actually attracts this initiator protein, so they can start replication. Okay, so the first thing for the DNA replication to occur is to open up the DNA. Remember, I mentioned in DNA structure video that the DNA is a double stranded helix. So in order for the replication to occur, DNA has to be opened so that this replicative enzyme can access the DNA, right? And the first enzyme in this process, which is also a rate limiting enzyme, is called the helicase. Okay, and helicase, what it does is that it unwinds the DNA. So it opens up the double stranded DNA. So now the DNA is a now single stranded. Okay, and it requires the ATP. Okay, so just imagine if the cell lacks the ATP, this enzyme cannot function and the DNA replication cannot occur. Okay, and this enzyme do its job by breaking down the hydrogen bonds. Okay, so as you can see here on the right side, the blue triangle serves as a helicase, and as you can see is moving towards the right side, and it is breaking down the hydrogen bonds. Remember, I mentioned in the DNA structure that the nitrogenous base are paired with each other via hydrogen bonds. So the helicase enzyme opens up the DNA by breaking down the hydrogen bonds. Okay. Okay, again, remember I also mentioned in the DNA structure video about the base pairing, the hydrogen, that the adenine and thymine are base paired via two hydrogen bonds, whereas the guanine and cytosines are base paired with, they are base paired with each other via three hydrogen bonds. And it is obvious, it's obvious that the breaking two hydrogen bonds are relatively easier and relatively low cost of ATP than three bonds. So cell will obviously prefer to this enzyme would obviously prefer to break two bonds instead of three bonds. So helicase enzyme prefer the AT rich region because they are two hydrogen, they have two hydrogen bonds. So they prefer to break two bonds instead of three. So as I said, this enzyme is opening up the DNA and it is breaking down the hydrogen bonds. But when this enzyme is functioning, there is an also tendency for these strands to reanneal with each other, because again they have a tendency to attract to they have a tendency to attract, so they can base pair again. But what actually prevents this reannealing to happen? So a specialized protein called a single stranded binding protein, it prevents the reannealing of the both strands. So this protein is very crucial. And as helicase is opening up the DNA, there is also tendency for the supercoiling, right? As you're opening as the helicase is opening up the DNA, there is a more chances of them to supercoil, which actually also prevents the DNA replication to occurs. And to prevent this, there is another enzyme which is known as a topoisomerase. There are two different types of topoisomerase 1 and topoisomerase 2. These two enzymes are very crucial and they prevent the supercoiling of the DNA. So this single stranded binding protein and the topoisomerase, they actually prevent uh, uh, they actually prevent the reannealing and the supercoiling of DNA strand respectively. Okay. Okay, now So in order for the DNA replication to occur, the first thing, there are actually various enzymes that are involved, and the main one is a DNA polymerase. Okay, and this DNA polymerase, they cannot start synthesizing the new strand from beginning. It needs some additional assistance from other enzyme such as primase. So the primase, what it does is that it adds the RNA primer to the DNA template. Okay, so the primer here, let me see if I can look at. So this is the primase which adds the uh, RNA primer, so that the DNA polymerase can start adding the complementary bases, right? And it actually opens up the door for the DNA polymerase 3 to add the complementary bases, right? DNA polymerase cannot function without the primase because it needs the initial assistance, and that assistance is provided by primase. Okay, and then once the primer is added, then the DNA polymerase 3 comes into the picture, and they start replicating the leading and the lagging strand. Okay, so here in the image on the right, the bottom one is a leading strand and the top one is a lagging strand. Okay, and as you can see here, as you already know that the DNA is an antiparallel fashion. So this is 5 prime, this is 3 prime, and this is 5 prime. And then so the DNA polymerase, it starts reading the parental strand from 3 prime to 5 prime direction. Okay, so it reads the DNA from 3 prime to 5 prime. As you can see here, it reads from this way, it goes this way. So it start reading from 3 prime to 5 prime, and it starts making the new daughter strand from 5 prime to 3 prime. Okay, so the most important thing here, here to note is that it reads the template strand from 3 prime to 5 prime direction, as I mentioned here, and it synthesized the daughter strand in 5 prime to 3 prime direction. Very important point to know. There are so many questions can be asked based on this concept. Okay, it reads from 3 prime to 5 prime, and it synthesized from 5 prime to 3 prime direction. Okay, and as I mentioned, it only adds nucleotide in 3 prime hydroxyl site. Okay, and this 3 prime hydroxyl site is created via this enzyme primase, because it adds the RNA primer. So it actually allows for the DNA polymerase to add nucleotides. Okay, and also one more thing to note here is that DNA polymerase 3, it has a higher processivity. Now what do I mean by processivity? The processivity means that once this DNA polymerase is attached to this parental strand and start making the new daughter strand, it has a tendency to actually hops off and prevent the transcription of sorry the prevent the replication to occur. So in order for to prevent this happening, there is another class of protein which is known as a sliding clamp, right? The sliding clamp is actually present there, and it prevents the DNA polymerase to hop off from the parental strand. Okay, so due to this sliding clamp, this DNA polymerase 3 has a higher processivity. Okay, okay. So far we've talked about all these enzymes that actually start making the newly complementary daughter strand. But as I said in the previous slide, there are two strands of DNA. One is a leading strand and another one is a lagging strand. So here on the right side of the image, the top one is the leading strand and the bottom one is the lagging strand. Okay, and the replication in the leading strand is continuous, whereas in the lagging strand it is discontinuous, and we will talk about why it is the case. Because just think about it. So as I mentioned before, that DNA polymerase reads the parental strand, the template strand, from 3 prime to 5 prime. Okay, so this is the 3 prime here and this is the 5 prime here. Okay, so DNA polymerase, once the primer is being added by the primase, it reads from 3 prime to 5 prime, and as this A is a helicase, as the helicase is opening up the DNA, and the DNA polymerase is actually continuously adding this base pair. There is no type of stoppage point here, as you can see here. So it can continuously add base pair until the helicase opens up the DNA. Once the helicase is stopped, then they cannot add the nucleotide. Okay, so in this leading strand, the DNA polymerase 3 actually goes in the same direction as the helicase enzyme, whereas on the lagging strand, the newly made strand, the newly synthesized daughter strand, it's synthesized as an Okazaki fragment. So they are synthesized in the fragments instead of the continuous strand. Okay, so because the reason for that is because, again, it is due to the different orientation of these template strands. So here, this is the 3 prime and this is 5 prime. Okay, so once the primers is being added, let's say the primer is added here. Okay, and it reads from 3 prime to 5 prime, so it reads in this direction, and it actually synthesizes in this direction. Okay, so the RNA polymerase start adding bases in this direction, as I'm marking with the red ink. Okay, it starts adding on this side, but by the time it reaches at certain point, let's say it reaches right here, helicase has already opened another piece of DNA on the left side. So here, as you can see here, the helicase and the DNA polymerase, they are moving in the opposite direction. So on the left side, the helicase is opening up the DNA. On the right side, the polymerase is start adding bases, right? So at one point, the helicase has already opened up the DNA. So again, another sort on another another primer has to be added, so the process can continually happen. The process can happen. So that's why the lagging star is synthesized as an Okazaki fragment, because of the uh, uh, opposite movement of the helicase and the DNA polymerase. Okay, and for the lagging strand, there is a multiple RNA primers are required for the lagging strand, because of the fact that they are moving in the opposite direction. Also, when DNA polymerase is making the uh, DNA, newly synthesized DNA, it also has a tendency to make a mistake. Okay, now this tendency is very rare. It happens, but it has a higher proofreading activity, so it can go back and correct the mistake. Okay, remember I mentioned it makes the new strand in 5 prime to 3 prime direction. So if let's say if it made a mistake, it has to go back from 3 prime to 5 prime to correct the mistake. So that's why they have a 3 prime to 5 prime exonuclease activity. Okay, because it makes the new strand in 5 prime to 3 prime. So it has to go back from 3 prime to 5 prime to correct the mistake. Okay, and again, the tendency of making the mistake is very less, but it has a higher proofreading activity. So it normally takes care of the mistake. Okay, now let's talk about the telomeres. So, what is the telomeres? Telomeres are a specialized sequence that can be found at the end of the DNA. These sequence are specialized, so they have a TTAGGG. This is the hallmark for the telomere sequence. Wherever you see this TTAGGG, that is a indicative of telomeres. Okay, and telomeres, as I said, is located at the end of the chromosome. So these primers cannot be actually added to the DNA at the end because of the steric hindrance. So the primers cannot attach itself to DNA, and since the primers cannot be attached, the DNA polymerase cannot do a synthesis, they cannot do replicative function there. So, and one more thing, the telomere is a functional gene. Okay, so if let's say the DNA enzymes cannot replicate, that means a cell is losing its genetic material, which a cell do not want, right? So what it does, the nature is very smart, and there is nature has evolved a process, or I would say an enzyme, which is known as the telomerase. It recognizes this telomere sequence. Okay, this enzyme telomerase, it recognizes this telomere sequence, and what it does is that it uses RNA as a template to make new DNA. Okay, and I will explain how this happens. So here on the right side of the image, let's say this is the TTAGGG. Okay, so this is the telomere, and the yellow is a telomerase enzyme. So it has an RNA as a template, as I mentioned, which is AAUCCC. It uses this RNA as a template to make the new DNA. So on the bottom, as you can see, this was the telomerase, and it added it uses RNA as a template, and it has made this new TTAGGG again. So now this one, this TTAGGG is preserved because it has just added a complementary DNA by using RNA as a template. So that's how uh, it has actually saved the telomeres from being not replicated. Okay, and if you recall, this telomerase enzyme has the same function as a reverse transcriptase enzyme that can be found in viruses. So what viruses do is that they use this RNA as a template to make their own DNA. Okay, so this telomerase has the same function as reverse transcriptase enzyme, and again they prevent the gene loss because telomere is a functional gene, and the cell does not want to lose the genetic material. And again, just think about it, this telomerase enzyme is very high in the cells that are continuously undergoing division, because such as like stem cells, epidermis of the skin, they are continuously undergoing cell division, so they cannot afford to lose the telomere. So that's why this type of cells that are continuously undergoing replication, they have a higher amount of telomerase activity and telomerase enzyme. So, and if this enzyme is lacking, then individual will start losing the genetic material. And also aging is also correlated with the uh, loss of these telomeres. Okay, and again, as I said, this is the different enzymes that are actually they are actually have the same function, but just the different naming in the prokaryotes and the eukaryotes. So origin of replication, as I mentioned, single. Eukaryotes has a multiple. Okay, polymerase is five type of, there are 14 different, you don't have to know these details. Telomerase is not present in prokaryote, it's only present in eukaryote. This is something that you would need to know. And primers is removed by DNA polymerase 1, whereas the eukaryotes, there is an RNase, and this is a polymerase 3. This is a different alpha, beta, gamma, delta, and epsilon. There are actually 5. And sliding clamp is just a different naming. And this is all the functional, this is all the function of the enzyme that I already mentioned. This is a good summary slide. Polymerase 1, it removes RNA primer and replaces with the DNA. Polymerase 2, it do the elongation in 5 prime to 3 prime. As I mentioned, helicase opens up the DNA. Ligase actually, when the replication process finishes at the Okazaki fragment, remember there is a still the gap. So this ligase, what it does is that it catalyzes the phosphodiester bond between the Okazaki fragments. So it reseals the Okazaki fragments. Primase, again mentioned, adds primer. Sliding clamp assists in the DNA polymerase. It helps hold DNA polymerase in place. Topoisomerase relieve the strain by supercoiling. And single-stranded, it prevents the reannealing of the strand, rewinding back. Again, the same thing. So that's it for today's discussion on DNA replication. In the next video, we will talk about the transcription. Thank you.